Welcome to this Winemaker Chat, a short video discussing notable results from the Winemakers Research Exchange. This format allows us to spotlight experiments that had meaningful results but were not presented during sensory sessions. My name is Ting, and I'm the research enologist for the Virginia Winemakers Research Exchange. I'm here today with Zach Pierce from Ingleside Vineyards to discuss an experiment Zach conducted in 2023. Welcome, Zach. Thanks for joining us.
Happy to be here.
Zach, this experiment focused on the timing of malolactic fermentation using co-inoculation and sequential inoculation of several of the red varieties. Before we kind of jump into talking about the experiment itself, tell us just a little bit about your red wines at Ingleside. What are your winemaking goals for that? Is there a particular style that you're trying to achieve with those wines?
So, I guess for me personally, because I've only been doing it this will be my third year, like at the helm, I'm still trying to develop my own personal style, but I prefer like fruit forward with like silky, smooth tannins, just well balanced red wines. I mean, really whatever I can get out of the vineyard. But I mean, we here at Ingleside, I mean, really the Petit Verdot is kind of only going to give us like a full bodied red. I mean, everything else is going to be much lighter and not going to kind of give me what I want specifically. So yeah, if that answers
And I
the
think
question.
you're not alone there. A lot of Virginia winemakers probably feel that way.
Yeah, I'm
So
slowly starting to learn that.
so the experiment itself was looking at using either sequential inoculation or co-inoculation for malolactic fermentation. Tell us, so in that experiment, what was your normal treatment, and what was the other thing that you were going to do, and why did you want to do that other approach?
So I guess for me, my normal treatment was the first year that I was in charge, and I was working with a wine consultant, and they basically said to do everything sequential. I didn't really get a good explanation why, but I mean, just being my first year, I just kind of listened to as much as they said as possible, and so that's what I did the first year, and it was just such a labor intensive process, and it was just it caused the cellar to be very congested. And then so when we thought about this experiment to see if it would really make a difference the following year, so that's when I was like, I want to see what the difference would be between co-inoculation and sequential.
So kind of a production efficiency kind of question, getting things
Yeah,
through quickly, not having to inoculate every single little thing and not having to wait so long. Is that, or were
yeah,
those?
exactly. I mean, and it's just another thing to worry about, like opening up and stirring each and every single barrel, having to make sure that they're all on one level. Yeah, so it's definitely a production efficiency thing
Yeah, yeah.
that I wanted to find out if it was worth it. If it makes the wine taste different, then we'll see.
Right.
But that was my biggest, not concern, but just the question that I had regarding it.
Yeah. Yeah. It's a great question. It's a great question. So I'm going to go ahead and share my screen at this point so we can look at some slides together, just sort of thinking about a little bit of background on that question, and then we'll jump into the setup and the results of Zach's experiment. All right. So Zach, do you see a screen that says microbial considerations on it?
I do, yes.
Okay, great. So there's just a couple of questions that, when we started to work on this together, there were a couple of questions that I started to look at in the literature that were kind of the questions I started to bring to the experiment as well. And the first of them is just, in order to do this experiment, we assume that we're inoculating for malolactic fermentation. And I realized that, I get to see a lot of people's protocols. Not everybody is inoculating for malolactic fermentation. So my first question was, why are we inoculating? What is it that, what's in that decision before we decide when to inoculate? One of them is just what are the things to think about if we want to decide whether to inoculate or not. So, if we kind of take a couple of steps back and just kind of look at the process itself, malolactic fermentation, is just this process of microbes consuming malic acid in a way that they release one carbon dioxide and leaving or lactic acid behind. So they don't break about the whole chemical. We just sort of take a little piece off, and we've got lactic acid at the end. And it turns out breaking that one little bond releases some energy, and that's what the microbes use to fuel some of their cellular processes. And it turns out that many different types of bacteria have the enzymes that they need to accomplish malolactic fermentation. Enococcus ini is the most common one that we find in the winery, and it's kind of the one that we prefer to find in the winery. First, because it sort of tolerates our wine conditions best, so it's most likely to finish for us and not kind of take a really long time to get done and sit around the cellar with unsulfured barrels for too long. But it also kind of produces the aromatics that we want in the wine. Unfortunately, Enococcus ini is, even though it's the most common malolactic bacteria that we can find in the winery, it's not the most common microbe on our grapes that can do malolactic fermentation. So the grapes come in with much larger amounts of lactobacillus and pediococcus than they do Enococcus ini. And the reason that that kind of matters is that even though these guys will do malolactic fermentation, they're also associated with a lot of spoilage characters. So things like acetic acid and ethyl acetate and mouse taint and biogenic amines. Those are things that are coming from certain types of lactobacillus and pediococcus. So even if so that's one thing to think about with regard to sort of inoculating, is part of inoculating is just trying to outcompete some of those spoilage organisms. But the other thing to think about is that, not all Enococcus ini strains are exactly the same. Much like our yeast strains are different from each other, the strains of Enococcus ini can be different as well. So there's about 1,700 genes in the genome of, expressed genes in the genome of Enococcus ini, and almost half of them have been shown to be kind of meaningfully variant, meaning that one strain versus another might have different tolerances, and they might have different aromatic profiles. And when we think about the aromatic profile of malolactic, sometimes it's like, well, does it really make a difference? And it turns out, at least in the literature, there are lots of different compounds that can be different in what's being expressed. So I kind of think about it like they all kind of have this set of metabolic systems that are like maybe the road system in your hometown. Rebecca and I both live in Charlottesville, and if we have the same roads to travel, but if Rebecca and I are leaving our office and going to visit a winemaker, I might be more likely to take one route to get there, and Rebecca might be more likely to take another route to get there. And in a lot of cases, that genetic diversity in our organisms is the same. They have all these same enzymes, but one of them might be a little bit higher affinity on one side or a little higher affinity on the other. And unfortunately, fortunately or unfortunately, that does cause some differences in the aromatic profile. So the thing we usually think about with malolactic is sort of the production of butter character or diacetyl. And we certainly see differences in Enococcus in how much diacetyl they produce and how much they consume, because they also consume it. But there's other things that also can change in the aromatic profile. There's esterases and ester consuming and ester making compounds that can affect the expression of red berry character. There are also some enzymes that can affect the release of oak lactones from barrels. So your malolactic bacteria can make it sort of more or less likely to get kind of a vanilla oaky character from whatever barrel you have it in. And also it's pretty well documented that malolactic reduces the amount of vegetal character in some of our wines, but the amount that it reduces it will be different between different strains. So whether you're inoculating or not, your Enococcus have the potential to do this, but if you are inoculating, sometimes you have a little bit more kind of choice along the way. I would say that the other thing there is, if you're not going to inoculate your mallow, do kind of be aware if you are in one of these situations where you're close to kind of the tolerance level of Enococcus, or if you have some compromised grapes, because at that point you may end up having a little bit more effect of those lactobacillus and this pediococcus. So again, that would be another time to maybe consider inoculation, even if you normally wouldn't do that. But even if you kind of do inoculate, sometimes that fermentation is slow and takes a long time. So maybe that situation, Zach, that you had last year, where it sort of felt like you were kind of waiting a long time for the mallows to get done, even though you had inoculated. And so the next question that I was thinking about is, why might malolactic fermentation be slow in that case? And again, it's really that much like our Saccharomyces, there are kind of tolerance limits for these particular microbes. So if we look through some of the main tolerance components that we have to think about, the first one's alcohol. So if the alcohols get too high, it does start to compromise cell membranes. It turns out these high alcohols make the cell membranes less fluid, but more permeable, which means that they're less likely to be able to get in what they're looking for in the environment. So nutrients and that kind of thing. But more permeable means they're less able to sort of keep their insides in and the outside out. In sort of a related topic, they do have a tolerance for certain tolerance limits for temperature. And if you kind of look at these numbers, these were provided by the Australian Wine Research Institute. These numbers it's kind of in the mid 60s is where they tend to do pretty well. And then there's kind of an upper and lower limit to tolerance. The lower one makes sense in the sense that if it's too cold, they just kind of can't run their metabolic processes as well. So they're probably not going to work as quickly. But then there's also this upper limit. And that upper limit really has two components to it. One of those is, again, has to do with their cell membranes. If they get too warm, their cell membranes start to not be able to keep the integrity of the cell anymore. So you can think of not only if it's warm, but also if it's warm and the alcohol is getting high, that's kind of a combined stress on the cell membranes that leads to inactivity and eventually it might even cause cell death. But then the other thing that happens when the cells or when the fermentations get warm or too warm, then basically our enococcus are less likely to be able to outcompete the other spoilage organisms. And at that point, they might have some inhibition from those guys. Another tolerance factor to think about is pH. And again, this is a pretty narrow range when we look at the favorable part of the pH between 3.3 and 3.5. And again, we have an upper and lower. And that upper limit, again, is mostly about outcompeting lactobacillus and pediococcus. We kind of know that when our pHs get high, we tend to have a little bit more spoilage around. And in that case, that's where that upper number is coming from. But then there's this lower number. And it turns out that's not because they can't tolerate that amount of hydrogen ions around. It really has to do with the interaction with SO2. So when SO2, when we look at sulfur dioxide, we know when the pHs get low, we have more of our sulfur dioxide is in that molecular form that's antimicrobial. And it turns out that our malic acid bacteria are really sensitive to sulfur dioxide. So whereas with like Brettanomyces, we need a molecular sulfur of like 0.6 or 0.8 or even higher to even inhibit them. With our malic acid bacteria, of molecular sulfur of 0.1, 0.15 is probably enough to inhibit their activities, and 0.3 or to 0.5 is probably enough to kill them. So we need to sort of think about the interaction between how much sulfur we've got in there and how low that pH is. But the other thing is that this is the one organism where the total sulfur actually matters. We talk a lot at the Research Exchange about how the free sulfur is really what's contributing to our molecular sulfur. That's kind of a proportion of our free sulfur. And so most of the time we say the only antimicrobial is coming from the free sulfur. But it turns out that with our malic acid bacteria, they're able, the main thing that's binding the sulfur is acetaldehyde. Malic acid bacteria can consume acetaldehyde, and when they consume acetaldehyde, they free that SO2 from its bound state and it becomes free. Once it becomes free, then a proportion of that becomes molecular. So in a way, by consuming the acetaldehyde, they're essentially slowly poisoning themselves with some of this SO2 that gets released. So when we think about malolactic, a lot of times we're like, well, I haven't added any sulfur yet because it's early on. But if you added some sulfur at crush, that became part of your total sulfur. So if you want to go through a robust malolactic, the less sulfur you have at crush, the more robust your malolactic fermentation is going to be. But the other thing that we need to think about is the impact of the yeast strain. So different yeast strains will produce different amounts of SO2 during fermentation, just as a byproduct of fermentation. There was one paper I was reading that looked at 4 different yeast strains, And the range of SO2 produced by the yeast during fermentation was between 13 and 42 parts per million. So that higher yeast strain, you can see it's already kind of getting into the range where just because of the yeast, even without any sulfur at crush, would start to inhibit the malolactic bacteria. So thinking about that, the yeast strain is also kind of one of those things to think about with a good malo. The other thing is about nutrients. So malolactic acid bacteria are not as good at getting nutrients from the environment as yeast are, and they need them in a different form than the yeast do. So unfortunately, we can't just run a nice YAN test and know how much we have to add. It's a little bit more complex than that. But we sort of, the general principle will hold that if you have kind of a good amount of nutrient around, you're more likely to be able to finish off. They're also very affected by pesticides and copper residues. So, so looking at this list, then the question getting back to co-inoculation, why would co-inoculation make this better? Why would it help us to have a more robust malic fermentation? And again, with co-inoculation, if you're adding sort of right after you add your yeast, then the alcohol hasn't built up yet. In general, your temperature probably isn't going to be too cold. We probably do have to keep an eye on the temperature to make sure it doesn't get too hot. Otherwise, we could kill those yeast before we actually give them a chance to do too much work. Here in Virginia, we're probably not going to get into that nice pH range of 3.3 to 3.5 on our reds. But if we think about that as kind of a range, the lowest pHs that we're going to see on our reds are usually in the must of the grapes, not after the end of fermentation, when we've had that big pH spike that happens in a lot of our reds. And then the total sulfur. So even if you've added at crush, those yeast haven't had a chance to add any more to that. So you're more likely to have good implantation of your malic acid bacteria if you have that sort of early inoculation along the way. So but then there's the sort of question of timing. So again, if we kind of look back at the different phases of fermentation, when we're talking about co-inoculation, we're usually talking about adding between 12 and 48 hours after after the yeast have been added. And again, another question I brought to this was, why do you have to wait? Why not just kind of stick them in there with the yeast and not have another step? And it turns out that piece is important. So the idea is that you wait until the yeast have just started to work so that they've been able to bind up any SO2 that you added at crush. So if you're adding at crush, you still have some SO2 around. And it turns out that if you add right away, you get kind of, I think it's like 100-fold reduction in the viability of your malic acid bacteria. So waiting that 12 hours to 48 hours, really just until you see some yeast activity, is an important step along the way. There are some places that think about doing this sort of mid-fermentation. And I will say that there was one paper I looked at that recommended that that's maybe not a very good idea. So what they found, again, was when they added in that kind of middle ground, they saw, again, they saw 100-fold cell death. And they said that, probably that's because the yeast they're most antagonistic at that point. So they're more likely to compete against the malic acid bacteria. You've got some alcohol that's started to build up. And at that point, the yeast have kind of eaten all the nutrients and there's kind of not a lot left. So it's really a bad environment for the malic acid bacteria. But then, of course, there's this sort of sequential or post-fermentation addition. At this point, you do have to deal with that alcohol. The wine's probably getting cooler as you go. It's not as warm anymore. But there is a big benefit to adding later on as opposed to earlier on. And that has to do with what happens when malic acid bacteria run out of malic acid. So when malic acid bacteria, they'll preferentially consume malic acid. When they're done with that, they'll eat up all the citric acid. But when they're done with that, if there's any sugar around and the pH is greater than 3.5, which it is in most of our reds, it will start to consume the sugar that's there. And the problem with the malic acid bacteria consuming the sugar is that that's when they produce really high levels of acetic acid. So there is this concern with co-inoculation that if your malolactic finishes before your fermentation finishes, then you can get these spikes in acetic acid. And I would also say that, even if your fermentation is done, if you're a person who has a lot of, let's say, whole cluster fermentation, and once you press off those whole clusters, you have like another sugar spike. You want to be careful if you've got some active malolactic bacteria that have finished the malic acid at that point, because that's when you might see an acetic acid spike. So one of the reasons that people might wait to do this as a sequential inoculation is that by that point, the sugar is gone and you won't have that spike along the way. So really, the co-inoculation is relatively safe as long as you have a robust fermentation, you keep on top of that and make sure that you finish on time, and also that you kind of don't have that situation where you've got maybe whole clusters or something else on the other side of it. So keeping those things in mind, let's go ahead and take a look at Zach's experiment. So Zach, tell us generally, how did you set things up with each of, we're going to look at four different kind of replicates of the same idea, but tell us generally how that got set up.
I just knew I was going to split it into either one T-bin per experiment or two T-bins per sample set. So it turned out I destemmed into I guess 4 T-bins for the Petit Verdot 2301 and 2302, and then another 4 for the 2303 and 2304. So that's how I separated the lots, and then everything else was exactly the same, down to the malolactic nutrients that I added. So everything was the same. So it got destemmed into the T-bin. Both got all of the other additions that I normally do: tannin, yeast, inoculated the same day. No cold soak or anything like that. The co-inoculated lots, those I did add the bacteria within that 12 to 48 hour range. It was probably somewhere in between like 36 hours or something like that. But yeah, I mean, that was basically it. I mean, it was a fairly easy experiment to set up, and then I just kept track of those T-bins all the way through until I had to press off, and then I separated them obviously into different barrels, equal barrels, same toast, same age. So each barrel as well. I think for these lots, it was 2 newer barrels from 2023 for each lot, and then 2 older barrels from like 2014, the exact same barrel. So I mean, everything was pretty much the exact same to a T, to kind of eliminate that variability. So
Right.
So yeah, I mean, that was basically it.
Yeah. Yeah. So just so you had 2 different Petit Verdot lots, and then we also followed the chemistry for Sangiovese and for Merlot. And for each of those, there were at least 2 T-bins, if not, some of them there were 4.
Yeah.
The sequential just got yeast at like the day of destemming, and then when did you add your malic bacteria for the sequential?
So for the sequential ones, they got pressed into a settling tank. They settled overnight, and then the following day, I racked them into barrels. And then I added the ML Red Boost. I either added the ML Red Boost to the settling tank when I had it all in a big lot, or I'm pretty sure I did that. I don't think I would have added them individually to each barrel. I could have, but it settled overnight, and then it got racked into barrel. And then after 24 hours after adding the nutrients, because that's what the book says, that's when I inoculated with the ML bacteria.
Okay.
So
yeah, so they got that. So they sort of got that nutrient and the ML bacteria later on, whereas the co-inoc got both of those things. Like, I think in your protocol was like 2 days after you did your initial
yeah,
inoculation.
yes,
Yeah. And just to clarify too, like we were adding the ML nutrients just to kind of keep everything the same. One of the good things about doing co-inoculation is probably you don't need to add those on a co-inoculation in a normal case,
yeah,
because you still have the nutrients from the grapes themselves. But you were very good about making sure that everything was the same between
yeah,
the 2 treatments, except for that timing of the ML bacteria. So it does give us a good comparison along the way.
yeah.
So just to take a look at our chemistry here. So the fruit chemistry itself, we can see that with these couple of different lots, it was great to be able to do several replicates of this. And we can see we've got kind of a wide range of acidity between the lots. The Sangiovese has a little bit more acidity. The Merlot has less acidity. We had different Yan types. And so we can kind of see what the capability of this is in different chemistries that way. I will say, like, if we were to kind of follow like the malolactic, the probability of a good malolactic for these, like the catalogs will give you sort of a calculator at this point. All of these would be considered to be pretty straightforward malolactic fermentations. None of these have any of the benchmarks of being super difficult malolactic fermentations. So then if we look at the fermentation kinetics themselves, I've just put the Petit Verdot on here. So Zach had 2 lots of Petit Verdot. They were both from the same fruit, but 2 of them got one yeast and 2 of them got a different yeast. So we're able to see how that looked in different yeast groups. And what we can see, really what this is showing us, is that we don't have any real impact of on the fermentation kinetics from whether or not we had malolactic bacteria. I think one of the concerns people have is sometimes that they might have some competition between the malolactic bacteria and their Saccharomyces in terms of getting their regular fermentation done. In an inoculated malolactic with Enococcus ini, these strains have really been selected so that they probably aren't making those competitive factors against Saccharomyces quite so much. And in this case, we definitely saw there wasn't any inhibition of the fermentation itself. And several of the papers that I read said that it didn't seem, if there was a slow fermentation, it did not seem to be because of the malolactic acid, the malolactic bacteria. They were going to either be slow whether you added bacteria or not be slow if you added bacteria or not. We weren't really able to get, there's not really like a good time to take a malic acid number here, because this also got a little bit of an extended maceration. So by the time these wines were pressed, they had sort of been on the skins, I think, for about 3 weeks. So, we probably had some malolactic fermentation that was going on even then. But we did get to look at the malolactic or the malic acids at the sort of middle of November. So that was about 3 weeks after the sequential ones had been inoculated. So again, the co-inoc ones were inoculated right after the beginning of fermentation, the sequential ones at barrel down. And so this malic acid number is kind of about 3 weeks after. And this is kind of about when, ideally these would be close to getting done, especially if you've done your inoculations. So when we kind of took a snapshot of that to see if these malolactic fermentations were done, we can see none of them were actually probably all the way. Well, actually, that's not true. So one of the questions that happens is like, what counts as done? And in the AWRI paper that I referenced earlier, they basically said that anything less than 0.05 grams per liter, which is shown by the red line here, is probably a good indicator of being done. They also said that anywhere lower than kind of 0.1 grams per liter was probably good enough to prevent spoilage moving forward. So if we look at kind of this lab test, we can see that the Sangiovese and the Merlot, those probably would have been considered done in both of the treatments. In either of the Petit Verdots, we sort of had differences between them, but in one of them, the co-inoc had more malic acid left, and the other one, the sequential inoc, had more malic acid left. So it doesn't look like we sped things up too much in this case. But again, in this case, they were both happening very promptly. And so we didn't it was going to be harder to go any faster than that either. Eventually, they did completely finish. Zach added his SO2. And so we went back in December and then looked at the basic wine chemistry that came out of these as well. And, the main thing that we were concerned about, again, was that acetic acid. So did we get into any kind of situation where we would see a spike in acetic acid with the co-inoculation? And if we look here, we can see, in the Sangiovese, we had a little bit higher acetic acid in the co-inoculated one than the sequential, and the same in the Merlot. But that pattern wasn't across the board. So in the first Petit Verdot, we had about the same acetic acid, and in the second Petit Verdot, we actually had the comparison switch there. So in that case, these differences in acetic acid may be as much just due to differences either in the fermentations themselves, because they were all separate fermentations, or in kind of what's happening even as they're starting to kind of finish their malolactics, just because they're in a barrel. And in this case, none of these is like super high or concerning, although I think we can see that. And I think right after this, you racked one of the Petit Verdot's that had a little bit higher acetic acid
Yeah.
accumulating. Um, so in that sense, you know, this seemed to be a pretty good approach at Ingleside, in the sense that it's not causing the things that are really concerning, and it may kind of help at least with that production efficiency, if not helping them out, the sort of Enococcus eni outcompete some of those other potentially spoilage organisms. So with that, Zach, you know, having done this experiment, you know, how are you thinking about co-inoculation moving forward? Is that something that you'll keep working on or keep trying? Is it something that, how do you feel about the results?
Um, I feel like previously, before I took over, that was basically, I think, almost what all of the red fermentations underwent was co-inoculation. And I think going forward, I mean, I'm probably going to just mainly co-inoculate unless I want to try something specifically like what you mentioned with I want to try like whole cluster fermentation, which I've never done yet. And then after what you just mentioned, it's like that might be the only time that maybe I want to do a sequential. But once again, that would be like experimental, like a much smaller lot. So I mean, for me, yeah, I'm going to probably do all co-inoculation just because it's that much more convenient.
Yeah, yeah.
Um,
So just before we kind of sign off, then I would say there's again, we'll review just a couple things to keep in mind if you're thinking about that. So again, you know, and there's a reason you followed the directions. There's a reason those directions are there, to kind of wait that 1 to 2 days before that co-inoculation happens, because that might help. Again, getting them in there early might help to ensure a complete and clean mallow in kind of a reasonable amount of time, really, because it's helping to outcompete some of the native flora that are coming in. And then, but if you are co-inoculating, again, the best way to kind of get the best result there is really to limit your SO2 at crush as much as you can, given the state of the grapes coming in. Making sure that you have some signs of yeast fermentation before you're adding your co-inoculated or your malic acid bacteria. Thinking about a low SO2 producing yeast, that was something that came up in some of the literature. And, you know, sometimes our catalog doesn't tell us this is low SO2 producing, but the clue that they give us is like, this is malolactic compatible. So like, if you look in the catalog and they're like, this is a good malolactic compatible yeast, usually what that means is that it doesn't produce a lot of SO2 of its own. But then the other kind of thing that came up when I was doing my reading was really that temperature maximum. And I do think that might be one of the difficulties with doing co-inoculation, is sometimes we have those sort of hotter fermentations as well. So just realizing that as the fermentation gets hotter and hotter, you're kind of knocking back that malic acid population too. So this probably is, but you were saying you're trying to make sort of fruity wines. Those tend to be a little bit cooler fermentations, and so it might be a good match that way. And then, yeah, just keep an eye on those fermentations, make sure that they're finishing well, because you don't want to have any sugar at the end for those guys to eat. So, okay, so any parting words before we sign off, Zach?
Um, I don't think so. I mean, we'll see. I mean, the thing that still intrigues me about just the whole malolactic part of fermentation, like maybe we could figure out an experiment as well, is doing when we do the acid additions, that malic acid addition, like or
Mm-hmm.
like I know we've done experiments in the past with it, but maybe doing the mallow with the tartaric instead of effects on, I guess, flavor, more so mouthfeel, because in my mind, I feel like if you add more malic acid, in my mind, I feel like that conversion to lactic acid should have a smoother, silkier mouthfeel. But I don't know. I haven't been doing this long enough to know. So that's
Yeah.
something that I might be interested in trying in the future. So,
Well, you know,
but other than that, yeah. Sorry, go ahead.
you know, if you say something like, I think I might want to do an experiment on this, I'm just going to call you after and be like, okay, let's do this experiment. So I'll be in touch. But I think that's a really good follow-up to this, to say, especially given the winemaking goals that you have for this wine, it might be helpful to kind of take a look at that and see if that would also help you to get to that goal. So so maybe we'll see that one next year. That's a great idea. So,
Hopefully.
all right. All right, so many thanks to Zach for doing this experiment and sharing it with us. And of course, we want to always thank the Virginia Wine Board for funding all of the activities of the Virginia Winemakers Research Exchange.
All right, thank you. Bye.
Bye.